Thanks for having me. I'd first likely to acknowledge the traditional owners upon the land in which a lot of my research has been conducted. I've been lucky enough to be across a lot of southeast Queensland and on beautiful country in the greater state. And I pay my respects to elders past and present. A great story I'd like you to all look up is the story of Wudjin, which is the Yugambeh language for platypus, the guardian of Scrubby Creek. And it's a lovely story about how platypus is very much that flagship species for our freshwater ecosystems. The platypus has definitely been an anomaly for us for many years. And right from when the first specimens arrived back in England, back in 1798, they thought it was a hoax. They thought, what is this weird duckbill beaver-tailed thing? It couldn't possibly be real because there were naturalists passing off different species by stitching different parts of animals together. So if you were to Google a mermaid, it was a monkey torso and then a fish tail. But they obviously never found any stitching within the platypus, and it just then created such an exciting time for back then the scientists to understand what this species was and where to actually put it, because it was egg laying. It also had milk, but not so, it didn't have nipples that it suckled from. It just exudes their milk. And so they put it off into its own taxa of monotremes, and the other one being here in Australia, the short-beaked echidna, and then there's the long-beaked echidna in Papua New Guinea. And it's well known for its elusiveness. Just because you don't see a platypus in the waterways doesn't mean it's not there. And that makes it really hard to detect and monitor. And so eDNA has definitely revolutionised the way that we can do this. But in a freshwater system, they have a lot to contend with. We're looking at habitat destructions along our waterways. We're seeing, the riparian zone being destroyed. We're putting a lot of concrete everywhere. We're seeing those hard and fast flows and those flashier flows within, especially our urban environments. And so there's a lot of impacts because of that. We then have the pollution as well, not just chemical pollution from runoff, but a lot of the hard rubbish, especially litter entanglement with hair ties and rubber bands, which is a huge concern, again, in our urban environments. We've got issues with fishing equipment. Queensland's still the last state not to ban opera house nets, where air-breathing mammals will die a horrific death in those, and we're wanting to swap them out with those open-top wildlife-friendly nets. We're then changing also the way water is used within our waterways, and we're having these big dams and weirs and causing massive barriers within our catchments. And platypus, that is a huge barrier in terms of these big dam walls, and it is causing complete disruption of populations from upstream and downstream. There is just no gene flow with things like that. In smaller weirs, platypus are pretty robust. They'll lock in and they'll claw themselves up smaller weirs, and they'll certainly try and scramble, but then they become a risk. It increases the risk with predation if they're not able to be safely within their water. We have native predators like birds of prey, but it's, as you can imagine, the foxes, cats and dogs that cause a lot of impact to our local wildlife. And platypus would never be able to outrun them if they were having to go over land to try and find water elsewhere. And also climate change, the frequency and severity in what we're seeing, especially drought for platypus. They really heavily depend upon water to feed, mate and move. And so that's a real concern. And then flooding, we have those severity and the frequency of our flooding that we're seeing is a huge concern as well. They're important in our ecosystems because they are a top order predator, preying down upon the freshwater bugs that they feed upon. So dragonfly larvae, caddisfly larvae, worms, mollusks and things like that. And when they are foraging, they're very vigorous in the way that they do that. And when they are lifting up plumes of the sediment into the water column, there's micronutrients into that, which will feed the microorganisms. So they're a part of that whole ecosystem. In terms of research, the female's milk has potentially got components to develop new antibiotics to fight superbugs. And then the male's venom has also been studied to understand potential proteins and things that can develop new painkillers, as well as a few years ago there was a potential cure for type 2 diabetes, which is quite exciting. We're not going out and milking platypuses for their milk or their venom. This would have come from captive bred platypuses, and then they can make synthetic versions of these. But overall, platypus are very iconic, and it's what we call a flagship species or an umbrella species. And so by promoting their conservation, we're ultimately promoting everything within our freshwater ecosystems and beyond, things that potentially people may not even think about, like the microorganisms and things like that. So back in 2016, the International Union for Conservation of Nature Red List increased their conservation status to near threatened. But and that was because of localised declines and a lot of research that has been coming out of New South Wales and Victoria. But that doesn't then, unfortunately, relate to our local Environmental Protection and Biodiversity Act, where here in Queensland, they're special least concern. So there is no conservation status. And a lot of that is because there has been a lack of knowledge to really understand and monitor populations. It's such a big state. We have platypus right from Cooktown, out west out to Carnarvon, right through to the border. So there's a lot of distribution here in the state to understand what is actually happening with them. So back in 2016, Wildlife Queensland, we wanted to understand, you know, the population and distributions within the regions, and we wanted to then also develop a survey design that can be repeated to build up that data, to build that consistency and that confidence in that data. And we're comparing that with a lot of the historical records, which has been traditionally observational records. Very rarely have we had population studies with netting and things like that. There's been some, but not a lot consistently over the years. So initially we looked at just the WildNet data, just visually looking at that data from 95 to 2005 and then 2006 to 2016. And you can see that there's a difference in the observations across Queensland within that time frame. And we were like, well, are we actually losing platypus? People aren't seeing them as readily, or were people just losing interest and actually not recording dots on a map that were really important for us? And so we wanted to jump on board and not only use the observational survey data, but we had environmental DNA come into the mix, which is absolutely exciting for such a cryptic species like the platypus. It does just tell us presence or absence data. As Mark said, we're not potentially looking, we can't understand individuals within a population. It would make my life so much easier if we could, because it's laborious trapping platypuses. But we can then start to understand species distribution, and then building up that data, we can infer some kind of abundance and then develop occupancy models with that as well. And then also model further some habitat suitability based upon those habitat features that support platypus populations. So we looked at the historical records across the region. This is more Greater Brisbane as well. And we certainly then went to local knowledge of like councils, catchment groups, to really understand where those platypus waterways were. And we looked at a lot of the sites and selected them on desktop based upon that data. And then obviously went out and recce'd those sites to see if we could get access and if they were suitable. Obviously, if they had water, some years were drier than others and things like that. So back in 2016, we started with 66 sites, and then we've been building upon that, repeating and expanding over time because of back then, it was obviously a fairly new application, and so we've built up over the years and had a lot more buy-in from our supporters through council and things like that to be able to expand the project. It's great because it's non-invasive. We don't have to harass the animals or anything. You don't even have to see the animals to obviously get a sample of their DNA in the water. As Mark said, he was talking about the multi-species analysis, but we were targeting specifically for platypus. And so part of their genome is that tells us that it's platypus is a mitochondrial gene, cytochrome B, and it's a 57 base pair fragment that we can pick up within the water. The lab then amplifies that. So they have the primers that then amplifies that in the sample to give us that presence or absence in the sample. And as Mark also mentioned, there's a gene bank. So there's a full library, and Sydney mentioned this as well, that, you know, there's a gene bank of many species, vertebrate species, microorganisms and now macroinvertebrates and things to do full biodiversity and multi-species analysis. There's been research into targeted DNA for platypus, and within 2 of the filters, so 2 of the samples, we're increasing our chance of detection up to 95%. So if they're there, we're hopefully definitely, well, by 95% confidence, we're going to pick them up in those areas. Obviously, there are limitations, though. Obviously, the way you set up the survey design, how many sites you actually have. One of our biggest limitations has been funding. To intensively sample along a waterway every couple of kilometres to really understand that distribution would be wonderful, but it's quite costly. There's also environmental variables as well. Obviously, the amount of water through the system can dilute the DNA. You've got temperature, you've got UV, which will break down those fragments of DNA that we're picking up in the water. So it still is a snapshot in time, but we're obviously wanting to, build and repeat on this to get confidence of understanding where those hotspots are. So yes, there's, with any method, there's always limitations, but we try to work out how we can certainly reduce that, and obviously building up a longitudinal survey and data has been able to do that for us. And it's as easy as passing through water through these 1.2 micron filters. We originally started with 0.22 microns, which was ridiculous and quite hard to push a lot of water through, especially if it's quite turbid. And then we put preservative through it, and then you can do multiple sites in one day, and then you can package that off and send it to Melbourne. So it's still a snapshot in time in terms of understanding where a species is, because that DNA will break down in that system. But it's pretty exciting because we've been doing this now for 10 years. We're in our 10th year building up this data set across Greater Brisbane, and we've had over 400 sites in that time. Not 400 continually repeated. Unfortunately, that we haven't had the funding to ever do that as such. But we've had some pretty consistent funding with a lot of our councils across the Greater Brisbane region to really understand and build up an understanding of where those hotspots are within the Greater Brisbane region. So one of the biggest years that we had was 2003, with over 300 sites sampled in the region across 88 waterways, with 6 local government areas supporting us. And 45 of those waterways were positive for platypus. And you can see certainly that there's, quite a lot of red around within the Bremer catchments, over in the kind of eastern part of Brisbane. Certainly the central part of Brisbane as well, and but we've got hotspots in the Albert catchment, the Upper Logan, the Reynolds Creek, which is Upper Bremer, and certainly along the catchments in Brisbane and South Pine River and Caboolture Rivers and things like that. So we're seeing consistent results over the region in a lot of these areas, which is great. Focusing in on Brisbane, we've got those hotspots. So in the north, Albany Creek, Moggle Creek, over in the west, as well as Pullen, Colo and Shelley Creek, and then the Walston catchment, Bullock Head and Sandy Creek here as well. Unfortunately, in historical waterways, this is Nogra Creek and Kedron Brook, and Cabbage Tree Creek. Unfortunately, platypus have most likely been completely lost in those systems, and that's been within the last 20 years. We do get a sighting every now and again of people saying they've seen a platypus in this system. And there, of course, our method of eDNA is never going to be, 100%. If there was one animal that was ever evading detection, it's still a functionally extinct population. There's not enough animals actually contributing to that DNA within that system for us to even detect it, especially if we're doing consistent sampling of multiple sites. We'd be pretty confident that we'd be actually picking them up in those areas, and we just haven't. So it's been a huge concern, and obviously quite sad to see that in a blip of an eye, 20 years, that we've actually seen platypus disappear from the Brisbane region. And part of my PhD, I published a small paper on that as well, and a couple of the Logan areas as well, that we certainly saw when we were consistently sampling, we weren't picking up platypus as well. Beyond that, I was also doing some rapid habitat assessments for platypus habitat as we were doing at these eDNA sites. And some of it related to their food source. So coarse organic matter had a quite weak association with platypus presence, and that certainly relates to them being able to forage through that coarse organic matter for their food source. The overhanging vegetation is not only just shelter, but the food source as well for habitat and things. Topographic wetness was quite strong because platypus need water, so no surprises there that it definitely needed that it was quite a strong association. And then complex benthic substrate, as our samples increased over the years, that became stronger as well. And then that relates to their food source. So this has been pretty exciting to build up this data over the last 10 years. And, you know, we can start to then delve further into analysis of determining that abundance and occupancy and finding those focused areas for rehabilitation. We can then hopefully delve further into more climate modelling and flow regime data as well, which has come out from Melbourne, that, you know, a lot of flow modelling very much influences platypus and their persisting populations. So we've got some certain, you know, further studies that we can do with this data and around the region. But it's been wonderful that the councils have backed this for so long, and they've seen the value of monitoring platypus over such a period of time, and they've developed management plans and specific actions based catchment by catchment to then, you know, rehabilitate those areas along the riparian zones or where there's potentially low connectivity in areas. And so understanding those hot spots has been able to then filter in to then direct those actions, which has been really exciting. And it all comes down to that connectivity of habitat, which again, it's not rocket science. All wildlife needs that habitat connectivity. So looking to the future, we're pretty excited because we have a regional multi-species project underway, and that's across from Noosa right down to the Sunshine Coast and out to Toowoomba. So it's going to be a big project, not just mapping platypus populations, but also, again, that multi-species analysis and that full biodiversity to understand the health of our waterways, especially those macroinvertebrates. They certainly are an indicator of freshwater health. And then we're really wanting to push out for a statewide eDNA survey and use citizen science, the power of citizen science, to help us do that. It's been over 20 years since the last census of platypus, and so it's very much overdue that we need to know what's happening in other areas of Queensland, because we have a lot of places that are data deficient from central Queensland and some places in the north. So we can harness this very exciting technology and the power of our citizen scientists to be able to help us to map a lot of this for us. So that's where we were hoping to go as well. This information then also feeds into that national conservation status for platypus. We're wanting to put in another submission because they are declining. The story is telling us that they're not doing so well in areas, but it's the data that we need, and it's the data that will then relate into that conservation status and hopefully that upgrade. So that's where all that data will go to as well. It's not just going to sit on the shelf. It is actually informing and doing what it can in the space of conservation for platypus and freshwater ecosystems. So thank you.
That was really cool. I'm just wondering about the cost of eDNA. This might be a question for Mark as well.
Yeah, so we use EnviroDNA and the samples for targeted eDNA, you're looking at maybe $100 per sample, so one of those filters, so potentially $200 per site. But the more sites you actually have, the cost reduces because they can batch. It's actually cheaper for them to batch process like quite a lot of samples. So if you've got a big project with a lot of samples, the cost does increase. If you're doing multi-species analysis as well, again, if it's just vertebrate analysis, it does go up in costs by $100 or more. If it's the full atlas, which is vertebrates, invertebrates, and microorganisms, you're probably looking at over $500 per sample. But again, you know, if you've got quite a lot of samples, the costs will reduce per sample as well. So it's pretty much case by case, really, depending on, again, as Mark said, what is your question?
Thanks so much. It was awesome. I just wanted to ask something I didn't cover, and it's just occurred to me, is transport of eDNA. Do you need to worry about that in your system? And have you got any idea of how far down, you know, the DNA is transported?
Because we're targeting specific species, we're working with platypus breeding season when we're actually going out and doing the survey. So we're hopefully increasing our chances of picking up the DNA detection because they're actually moving in the system a lot more, and hopefully contributing to that DNA. And we assume, and as you know, depending on the system, we're probably, again, and from EnviroDNA's, like they've done a lot of background kind of validation and things to the eDNA, but you're probably, if at a site on a waterway, you're probably picking the DNA up maybe 500 metres, maybe on a good day, depending on the flow and the temperature and things, maybe up to 1 kilometre, but that might be at a push. And so intensive sampling, if we had all the money in the world, we would try to probably do it every couple of kilometres along a waterway, to pick up, you know, that more fine scale distribution along waterways and within catchments. But unfortunately, we just, the biggest limitation for us has been the funding and to have that many sites around the area. So we've really just had to work with what we've got. But we've been lucky enough to certainly build that up and repeat sites. And so, when we go back and repeat and we continually get positives in an area, you know, we can certainly say that they're within that area, they're persisting in that area. If we get years where, like, it was positive one year, then it's negative, and then it might be positive again, potentially you've just got a transient animal through the system. We've just been lucky enough to pick it up within the days that it's passed through that system. It may not be holding out. So again, there's a lot of those variables depending on the species as well.
Thank you, Tamielle. Awesome job.